Hi, so my name is Jensen. I'm an extension agent from Oahu. I work with edible crops, so fruits, vegetables, herbs, spices, beverage crops. I think that's all of the edible crops categories. This project for me started 3 years ago. A couple of the wholesalers were coming to me to ask if we could get mangoes in the market when the mainland market and the South American market stops. And so based on some old publications on forcing mango trees, I took on this project to try and get mangoes in the winter for some of the varieties that are preferred. And so that's how this particular project started. So some common data, I guess, at least from what I could pull. So harvest days are from June to August, October for different varieties are some of the normal harvest dates for a Hawaii mango, the common Hawaii mangoes, I guess. And then what I found was California season is from July to September. The Mexican season is from January to October. So after October, mangoes typically stop production or at least importation. And so it was trying to figure out a way to get mangoes outside of October into December was the whole point of the project. But as we went along, so this project took place in Waimanalo, which is a pretty rainy area during winter. And that's typically when the trees are flowering. And so we lose all the flowers and we have no mangoes ever on those trees. And so the project kind of turned into a powdery mildew anthracnose mitigation project on top of that. And so I won't go into too much detail because I think this is going to go into more of that. But yeah, so powdery mildew is one of the diseases that will wipe out the flowers. And then you have anthracnose too, that can decimate the flowers. And so in the rainy period during flower initiation is kind of when you lose a lot of those flowers and then you don't have any fruit set. And so we're having that issue out in that field, which helped me because there was no flower or fruit on the tree so I didn't have to get rid of them, so that we could force the tree later on in the year to get December fruiting. And so this is kind of what I based this project on, this kind of model. So there's different factors that can initiate flowering in a mango tree. So chilling is kind of one of the main ones. So during the cooler seasons, it'll initiate the plant to think it's time to flower. If you defoliate or pruning, you put stress on the plant, that could also trigger flowering. But I want to focus on this nitrogen category, so calcium nitrate, potassium nitrate, ammonium nitrate, and then this new project that I'm working on now is sodium nitrate, which is an Army approved product. So these three nitrogen sources are not Army approved, so you can't use it for organic production, but sodium nitrate has an Army approved product. And so you could use that to trigger flowering off season. And so if you look at the induction period, that's kind of the area that I was focusing on. So initially when the project started, we had to try and prevent flowering. So one of the triggers is gibberellic acid. So GA3 here on the left, you could tell the plant to skip flowering and stay vegetation. And so the idea would be originally was to treat your tree with gibberellic acid 3 in say January, February before the flowering period starts. And that triggers the plant to not put flowers out during that period. And so the plant will go vegetative. And so it'll take in more energy so that you could force it later on in the year. It would have the energy to put into the flower and fruit later on in the year. That was the original part of the project. But because we lost all our fruits, all our flowers originally, I didn't have to spray the GA3. So we could save a lot of money during this period because we didn't have to spend the money on the GA3. We just let it naturally fall off. And then we can come back in later on in the year when it starts drying up and then spray the potassium nitrate or the nitrogen product to force the flowering. One of the key things that I found is that you have to make sure that the plant is ready to push out the buds and has not already initiated bud break. That's kind of the period. So the buds have to be kind of like A, where it's starting to swell up, but hasn't really opened up like C through F. That's kind of like the period where you have to treat it so that it triggers the cells in that tissue to not be flowers or to be flowers. And that's kind of like the idea. If you spray it too late where it's already going, it's probably not going to catch it. You're not going to get flower initiation. Or if you go too early, you might you might not get it at all either. So you have to kind of like watch the buds or know when the buds are starting to mature typically during the year, and that's the kind of time you want to spray it.
So that you sprayed it with type of fertilizer.
Yep. On the foliage. And so the first rates that I went with was 15 grams per tree of potassium nitrate in 500 mils of water per tree. And I use this organic surfactant called Kinetic. It's a penetrating surfactant, so it's supposed to help get the chemical into the plant tissue better so that it can absorb it better. And then that first year I sprayed gibberellic acid 3 just to see if I could prevent the trees from flowering.
How's it going?
And it worked pretty well at preventing the trees from flowering. So the first round is we're normally getting flowers on our trees from February to March. And then that harvest period would be July through August for those trees. That's the normal flowering period. And then we had a treatment of potassium nitrate to see if we could increase flower production during that same normal period. So at flowering time, I would spray them with potassium nitrate to get more flowering, but it didn't seem to get any more flower than the control trees. So it wouldn't be the best time to spray. It's a wasting fertilizer, I guess. And then what we did was force the flowering in June through July with the potassium nitrate to get flowering and fruit in October through November for this first round. So this picture on the right is the gibberellic acid that I use to prevent flowering. And it works to stop flowering if you don't want flowers on the tree. And it helps so that I didn't have to go in and prune all the old flower stems off the tree because there were no flowers from the gibberellic acid. I use a backpack mist blower, for this particular project. And for bigger fields, I've come up with a modified version. So instead of having 4 gallons of water on your back, you just get a regular leaf blower from whatever brand you want to use. You can connect it to a spray tank. And all you have to do is buy the nozzle from the mist blower and attach it to the front of the leaf blower, and then connect the inlet for the nozzle to the hose tank, or the hose from the tank.
And then does that pull the liquid out of the tank by Venturi? Or
No, so you need a pump. You don't need a huge pump like this one, this high powered pump. You can use a battery powered peristaltic pump or diaphragm pump. All you need is like 30 psi of pressure probably to get the water from the tank to the nozzle. It's so the liquid coming out is just a little bit of push from the tank pump, but the dispersion of the product is with the blower. So it'll atomize the liquid coming out slowly from the pump, and you'll get really good coverage at up to 20 feet if you have this bigger model. It's a 450 or 450, I think, or there's a 470 or a 370. So the 370 is a little bit smaller model, and it can't really push up as high, but the bigger model can get up to about 20, 25 feet.
And you have valve in line there too, just
Yep.
above.
Yep. So on this one, there's a more of a shutoff valve right here. And so I can deal with that if I wanted to, or my newer models have like pressure gauges, and then we can put pressure reducers on top so it doesn't exceed a certain amount of pressure coming from the tank, so you get a steady flow of liquid all the time. Most of these sprayers have a orifice nozzle, so this nozzle actually determines how much liquid can come out from the spout, and so that'll help you control the volume of water going up into the tree too. So prior to application, there's some flower initiation already happening during this first round. So I had to cut all of those off to keep them all consistent, but there wasn't that many. And so the tree on the left was treated with gibberellic acid, so there's no natural fruit flowering from that particular tree. So the GA works to prevent flowering if you want to do that. And then you can see pretty much no difference from the untreated trees and the trees that got potassium nitrate during natural flowering. So there was no real increase in flower production and fruit set during that period. This year had a little bit drier year, so we didn't have as much fruit loss. In the left was then treated with the gibberellic acid, the potassium nitrate. So you can see the forced flowering happening in July. And then the trees that had natural flowering or additional potassium nitrate, you can see the fruit starting to mature and ready for harvest during that normal harvest period. And then in August, you see pretty much most of the fruits have been harvested or are about to be harvested off the tree. It's the end of the harvest season. But you can see the trees on the left that were forced in July are basically still have developing fruit. September, basically no fruit on the originally treated trees. And then the delayed treated tree on the left, you can see getting closer to ripening. And then in October, we have almost ripe fruit ready to harvest. And so you can get the fruits out into October if you wanted to, using a potassium nitrate or a nitrate-based fertilizer to stimulate the flower initiation during off-season. And then I wanted to take it further into the year. And so we did a little bit later treatments out until July 27 was our last potassium nitrate treatment so that we could get fruit in December and January. So the same, this one I increased the rate to 30 grams per tree with a little bit more water because the trees are getting a little bit bigger, so I could get better coverage with the solution per tree. That's why you see a little bit more volume of water. And it's based off of volume of weight of product per volume of water. So every 500 milliliters of water, I put 15 grams of the fertilizer. And so the more water I put, the more potassium nitrate I had to put in the tank just to get the concentration right on the tree. And then the surfactant stayed the same, which is the kinetic. And then, so this is a Raposa field that I've been doing all this work in. And so what I did was I randomly scattered the treatments throughout the field to make sure that there was differences. So this field actually started as a density planting field. So the first row at the top, I think it was like 8 feet spacing or something like that, all the way down to 20 feet spacing, I believe, which is the 3 trees. And so I wanted to make sure there was distribution of the treatments in that field so that it wasn't swayed by the density of the planting. So this is the second year on those same trees that we treated the year before. And so it's, sorry. The treated trees, you can see this was treated in June on the left. And then the normal trees that were being harvested that were untreated on the right. Sorry, I'm mixing that up. So these are treated in 5-25. And then you can see in July, they're golf ball size fruit. And then by September, they were pretty mature size fruit. This is what the flower looked like in June. I'm mixing these up. And these ones were treated in June. So you can see the flower initiation in July, fruit set in August, and then fruit development in September. These ones are treated in July. So August, you have the fruit initiation, and there's some natural fruiting left on the left. But then you can see the flowering maturing in September. And then what I wanted to do was this year was a rainier year, and so the trees that were not treated at all had almost no fruit because most of them fell off at the flower stage. But with some kind of treatment throughout the year, you got at least 60 to 80 fruit on the tree. So you got an increase in fruit production just by forcing them outside of that rainier period. So my current recommendation for this project is if you are in a rainier area, then just let the flowers all fall off from the disease if you do get it. And then you can come in and spray from June through July, and then you can get later fruiting. And then this year, after this particular trial was done, I came in and actually whacked all the trees. So my boss wanted the trees pruned, and she didn't say how she wanted it pruned. So I kind of took it severe. So this is what the trees look like after the project before pruning. So kind of when harvest was done, I came in, and this is what the trees look like after pruning. I kind of got overly excited with the chainsaw and went at it. She wanted it shorter, so I made them shorter. And then it made me want to see if I could force the flowering after 1 year of growth. So that's why this project came about, or this current project came about. So you can see a month later, there's the leaf initiation coming. A couple months later, in April, you see there's some flowering happening. And then in August of this year, this is what the trees look like. There was
That was 1 year.
This is 1 year of growth on those trees after that severe pruning. And so I wanted to see if I could get fruit on that 1 year of growth so that you won't lose a season if you prune it back that severely, basically. I also wanted to include this sodium nitrate, which is the product name is called organic. It's about the same amount of nitrogen as potassium nitrate, about 15% nitrogen. But it's an OMRI approved, it's like a mined sodium nitrate. It's water soluble. So I went at the same rate, but I put 2 liters of water so I could get better coverage. Same surfactant with the kinetics. So for an certified organic grower, you could use this material and you should be okay with your certifier because they're both NOP compliant, OMRI approved products. And then I want to compare that to the standard, which is the potassium nitrate, to see the differences. And so in August, about 3 weeks later, you can see flower initiation. I noticed that when you spray these trees with a nitrate, you get these funky leaf growth too. If it's like already decided to be a leaf and you force it, it puts out these like weird elongated, I guess you can't see the mouse on there, elongated stems, which think they want to be flowers, but don't have any flowers, which is kind of funky. And then in the orchard, instead of randomizing, I just took a row because it was easier to spray. So the row on the right, on the photo on the left, is a sodium nitrate row. There's an untreated row in the center, and then the left is the potassium nitrate. So the potassium nitrate has way more flower initiation, I think, than the sodium nitrate, but there is some initiation with sodium nitrate. And so I'm not sure if I have to increase the concentration. So that's a future project to work on, is getting the rate of the sodium nitrate down, because it may not be as plant available as the potassium nitrate because it's a mined mineral. It might need further breakdown or a higher concentration. But it did show signs of initiation. On the 19th of September last month, we had pretty good fruit set. They're about marble size to some golf ball size. And then just yesterday, 2 days ago, I took a photo of the fruit size, and they're about golf ball size now on the trees. So this photo on the left was that heavy flowered tree, and then the one on the right is the sodium nitrate. It's a little bit hard to see, but there's maybe about 15 fruit on the sodium nitrate tree versus maybe 100 fruit on the potassium nitrate tree. And so, yeah, so far, we haven't taken it to maturity yet, but so far after 1 year of growth from severe pruning, you could force your tree into flower production and have fruit for that next year. So you wouldn't miss a harvest season if you did it that way. And then the trees will stay pretty compact, easy to harvest from. So these trees now are probably 12, 15 feet tall. And then we've done it on a whole bunch of varieties. Heat, Namdak Mai, Austin, Govea, Fairchild. So Fairchild had, to me, the nicest flowering, but the other trees had like partial initiation. It could have been just the timing of that bud maturation, because this is all done in August. And so those trees might not have been mature enough to push out the flower buds, versus this Fairchild might have been ready to shoot out buds. And so that's why it had heavier flowering. But so far, the only trees I haven't got to flower were the trees up here, which we tried last year. And I don't know if because they were too tall, I wasn't getting the chemical high enough with the small sprayer that they had. Could have been the problem, or because they're taller and bigger, maybe I needed a higher concentration of the nitrate product. But so far, it seems to work every year, and you can just keep flowering it. Those trees too had fruit on it, and they haven't died yet from being sprayed from this year. So I think you could I wouldn't suggest spraying it more than once a year, but you could probably get it to, you could even spray half the tree probably, and then spray the other half later. So you have fruits on the same tree different times of the year.
It's a fair trial of a naturally heavy bloomer,
That I don't know.
but I know it's some.
Yeah.
Some varieties are heavy bloomers compared to others.
Yeah.
Like several fruit sling that was really like heavy bloomer and said it will everywhere. I used to not flower, not
Fruit,
soft,
because there's too much.
so
Yeah.
and it said, well,
Mm. So that's another opportunity too, is if you have poor flower initiation on your tree that year, you can always come back in and spray it later on to get those, because the tree hasn't used up all its energy for that fruit load. You could probably force it again to be able to handle more fruit on that tree. And so moving forward, this one now, because it's probably going to fruit in, say, December or harvest in December, it's kind of evaluating the fruit now to see if the quality of the fruit changes because they're getting into shorter days, less sunlight. So is the sugars going down or the fruit quality getting, size getting smaller because it's not going through the longer days of the year? Those are other questions I have that don't have answers to. Because then it'd be worth it if you have junk fruit in December to put all this energy in. So that's the next stage of this particular project, is figuring out if it's good quality fruit now in December.
comparing it, I mean.
At that time, yeah,
Consumer,
yeah.
you take it or.
Yeah.
You leave it, so.
I had oh, what was the question?
Oh, whenever there's questions in the room.
Oh, okay, I can repeat the question. Sorry about that. Yeah, so I think anecdotally, the last time I did it was there was a December harvest, and it was just people going into the field and picking what was ripe at the time. And so I haven't had any negative comments from them, and so it must not be that bad. But there's no quantitative data to prove like bricks readings or actual weight of the fruit. And so this year, that's my plan, is to see if there's so weigh these fruit and compare them to historical data for the fruit weights. And probably next natural season, or go harvest other Raposa fruit and then take bricks readings to figure out if there's any differences in the bricks readings for the sweetness. And just some summarizing comments about forcing flowering on mangoes. Is there? Oh.
Thank you.
Yeah, so the main purpose of this project was to get that winter market that nobody else is doing, and try to figure out an economic way to do it. And 60 grams per tree is not a lot of fertilizer to use. And so cost-wise, it's the labor to spray the trees with the product. And if they're already spraying fungicides on the tree, maybe this is a cheaper way to get one out of the disease period. So you're not spraying fungicides, you're spraying a cheaper fertilizer product, and then you're getting into a later market, which has a higher value. And so your economic return is going to double by doing that practice, hopefully. That's kind of the main objective for using the off-season flowering. I would have to calculate how many trees per acre to figure out like a cost per acre basis. But a 50-pound bag of sodium nitrate is probably $120. And so you could spray a few thousand trees with one bag. Yeah. Yeah. Yeah, it's like maybe an eighth of a teaspoon per tree. From a volume standpoint, 60 grams is the most I've been spraying per tree. And then the kinetic product is, you don't have to use it probably, but I haven't tried without the kinetic. So that's another trial I could do too, is with and without a surfactant. But I just had the product and figure I might as well put it in to get better absorption of the fertilizer to make more use of it. And then, yeah, if you already have the spray equipment because you're spraying the fungicide, then there's no additional cost for another spray. There's no questions online. Is there any other questions? Okay, thank you.
Thank you. Yeah, okay.
All right. Thank you, Jensen. Our next presentation will be from Lisa Keith. All right. It's these direction back.
Okay, hi, everybody. I'm Lisa Keith. I'm a research plant pathologist with USDA Agricultural Research Service in Hilo, Hawaii. And we're going to talk about the mango research that we've been doing for the last few years. It's been an awesome collaboration between my amazing team that you see, Lionel, Marianne, Danny, and Caitlin. Ho'omalu Farm, who's actually an organic mango farm, and the team here with Andrea and Matt. Yes, yep, the Wakefields, yep. So as a plant pathologist, I kind of broke down today's talk, a chance to just give some plant pathology wisdom and nuggets to start the talk, pretty much how disease is caused. What is a disease cycle? And how important it is to actually prove pathogenicity of something new that we find in the field. Then I'll go into fungal attackers of mango, give our overall research update, and then end just like Jensen did with the summary and future plans. Okay, so plant disease is caused by a biotic or infectious agent, and it's a process or change that occurs over time. Okay, that's different from something abiotic. It's different from, say, a lightning strike where suddenly you see something, but it happened immediately. It happens over time. And it's important to understand disease by, first and foremost, what is normal for your tree. Okay, is there a color change, you know, looking at the fruit or the leaves, or if there's death or wilting, is that something that naturally occurs, or is that contributed to disease? And when we talk about plant disease, we talk about the symptoms, which are actually how the host responds to pathogen attack. So fungal attack causes then the symptoms. And then there are also signs, the physical presence of the pathogen causing the disease. So maybe seeing discoloration in wood, which is because you're looking at the fungus in the wood. And I mentioned that it, especially for first time occurrences of maybe a disease in our area, you have to prove pathogenicity. Just because you find a fungus and can get it off the fruit doesn't necessarily mean it's causing the disease. Okay, a primary pathogen causes the disease, but there are pathogens called secondary or opportunistic. Oh, there's a wound, and maybe it gets in, or maybe the disease is happening and something else in the environment gets in. We may actually isolate it, but that's not what's causing disease. Okay, so just quickly, with plant pathology, we always talk about the disease triangle, and that's how disease happens, with all parts of the triangle occurring at the right time and the right combination, needing the pathogen, and in this case, we'll be talking about fungi, a susceptible host, and then the environment. Seems very simple. It's actually very complicated. And when you're even trying to recreate this in the laboratory, the greenhouse, it's very difficult. Disease has a cycle. You figure The spores of the fungus have to be produced. They have to get to the tree. They have to get inside. They have to do what they're going to do to cause the disease, multiply. Is there an alternate host? Or, if temperatures are too cold, maybe where do they overwinter? And the process will continue, and how many times per season. And I mentioned proving pathogenicity. Well, there's a set of steps called completing Koch's postulates. This was scientific back in the late 1800s, and we're still using it today. And Jensen already started going over the two main problems that we have here with mango in Hawaii. Powdery mildew, there's the fancy name of the fungus, Oidium. It is widely distributed throughout the Pacific. Wind disseminates the spores, okay? And it's more common in lower rainfall areas, but it does occur yearly. And so again, the symptoms, how the host responds, it can attack all portions of the tree. And you'll see kind of a grayish, powdery looking material on the new growth. And usually the flowers will abort and things will fall off. As far as control measures, well, it's possible to use less susceptible cultivars, some cultural practices, optimize nutrition, and use fungicides. And the other one is anthracnose, being caused by a variety of species of the fungus Colletotrichum. And again, the spores are splashed by rain, high humidity, warm temperatures, promote disease, encourage spread. You see necrosis on the leaves, so spots, dark spots on the leaves, the branches, all tissues. And then not only can the disease start in the field with the fruit, but then it becomes a post-harvest issue. And it's a lot of pruning. Now you see amazing pruning options, right? And removing that material. You don't want to just prune infected material and leave it in the field, but if you can increase airflow, helps the moisture not to be contained in the canopy. And then there's treatments as far as available for use like fungicides. But the challenge and what this grant was, it's actually a multi-state, multi-researcher grant for the organic side. And it even made the paper when it was first announced. Can essential oils be used to fight mango fungi? And so the granting agency, NIFA-OREI, and that was essentially plant safety, hort benefits, disease efficacy of essential oils for organically grown fruit crops. And it mainly led by the University of Florida with collaborators at Clemson, Georgia, UC Riverside, here at UH, our lab. And we were responsible for looking at mangoes, but there's also work on avocados, blueberries, and peaches. Many objectives for this multi-year grant. The first being, well, evaluate the safety of these products and see in the laboratory if there's any potential. I mean, if there was none to start in the lab, it wasn't worth moving forward. And this was done on the mainland, looking at things on plates, things with fruit, potted plants. And then from there, it went to, well, is it effective on farm? What does it do in the field? And then objective 3, what does it do post-harvest? And then 4, of course, to dispense as much information as we can to our growers. So when we got started, we had isolated quite a number of collies, Colletotrichum. We wanted to make sure that they were in fact virulent, meaning they can cause disease, and well identified. I can talk to you all day on the difficulties of, figuring out what the names are. But through the partnership, we sent a number of cultures to Florida. They identified them. There you see the 2 species. They're common pathogens, not only mango, so lots of hosts in warmer tropical regions. They wanted to assess the aggressiveness is what you're seeing on the fruit and the different life stages on the left. That's the fungus growing on plates. And then what do they do? And were they susceptible to the products on plates? Okay, so the two products, one's called Thyme Guard. That's the essential oil of thyme. And Thyme-X was a combo of thyme and savory oil. And really just what you're seeing is, okay, if you amend the media on a plate and you put the fungus and it starts to grow, can the product inhibit growth? And so the bottom left, you know, after a number of days, the fungus will colonize the entire plate. Well, what do the products do and what kind of concentration do you need? I mean, if you're already needing above label rate, well, it's not, I mean, they're not going to change the label for our application, right? So that was just some of the basic preliminary rates. The work, I'm sorry, Thyme-X, sorry, tea tree oil. I'm getting confused with another product. Tea tree oil and thyme oil. Long story short, Thyme Guard showed promise and Thyme-X did not. However, as they were doing things in the laboratory, we started in the field to see, well, new products, what is the potential for some kind of phytotoxic effect? And this was, again, working at Ho'omalu Farm with two varieties of mango, Raposa and Manganiello. And then, you know, the fancy, yes, random, labeled, and using low and high rates, weekly sprays on all parts of the tree to actually observe, was there any phytotoxic effect? And it was good news. No, we saw no phyto in the field. And then the research continued, and I've tried to summarize and, you know, happy to answer questions or provide information. Again, it was a randomized design. Individual trees were the reps in the field. We would tag branches and look at the panicles, look at the flowering as they were occurring, actually with then these treatments, comparing Thyme Guard, a potential new product, with the grower standard practice, which was using a combination of sulfur and copper. And sulfur is applied only on the flowers and buds as they're starting. And then copper is applied essentially once a month until fruit harvest. The products are sprayed until near runoff. And we would then go and take all of our data. Okay, so for 2022 and then 2023, there were 4 Thyme Guard treatments versus the 6 copper sulfur the first year, and then 4 versus 5 the second year. Severity rating, so we're looking at what percentage is infected, okay? Using a visual rating scale for all of the tagged branches. And the first spray for the first field trial occurred December 21, the second in January of 23. And then we would evaluate every 2 weeks, the collective team taking lots of data throughout the whole growing season. So that was the field results. And so for 2022, definitely we saw variety effects. We saw the seasonal effects. For both products, there was the overall increase in the powdery mildew. Little to no anthracnose being seen on these panicles over time. But as you heard, even with Oahu, there was something crazy going on with a weather pattern that really amounted to very little fruit in 2022. And strangely enough, it wasn't just Hawaii. Florida said they virtually saw almost none in their field trials, which they had, you know, 30 years of research. They had never seen that before there either. So definitely something strange going on. To summarize 2023, we got then the percentages where how many panicles would have fruit, and what was the panicle rating for anthracnose, the severity. And so there are the numbers, and I tried to summarize on a graph with the various, so the 2 varieties and then the 2 product combinations. And long story short, you put the stats on and it pretty much shows the same thing. They were both equally effective. Okay, so showing some promise, and especially really with copper having that blue coloring left on the fruits as it's moving into the season. That was maybe one positive. That Thyme Guard, you see nothing. Yeah. Okay, so we were paying attention to what was happening in the field. And then at the end of the seasons, we would collect fruit, actually bring them back as shelf life studies. And so the fruits were harvested and packed by our growers. The boxes would contain anywhere from 4 to 10 fruit, back to the laboratory, held at 21C. For 14 days, constantly taking ratings as day 0, essentially what the fruits look like coming from the field. And then every few week to 2-week period, using the visual rating scale of severity again. Okay, so for 2022, the summary of data, you can see the harvest dates in July, with the 2 varieties, again, the 2 treatments. Day 7, you really don't see much difference. Now, as it's held to day 14, that was a different story. Anthracnose, the symptoms we saw in all fruit, regardless of what was being treated in the field. Okay. Now, I will say that right now, fruit are never going to last 14 days. I mean, you know, they are immediately brought to market. People buy it immediately and eat the fruit. This is really looking towards, well, is there a possibility of potential shipping or holding on to, you know, if by chance, but again, up to about a week, both treatments, the grower standard and the potential new product, looked pretty good.
Excuse me.
Oh, yeah.
Temperature were they held at?
21C. Yeah, so 70, I think, about 70 degrees.
So, kind of lower room temperature.
Yeah, yeah. Okay.
So if you can read the question.
Oh, I'm so sorry. So what was the temperature of the experiment for the post-harvest studies? That was 21C. I'm pretty sure that equates to about 70 F, but yeah, a bit of a slightly chilled temperature. Yeah. Here are the results of the 2023 post-harvest. And so what we had 3 different post-harvest trials. We combined all the data based on time. Again, you know, the various colors, it's the varieties, and then the control is the field treatments of the sulfur copper, and then versus the Thyme Guard. And so you start to see a little bit of difference. Day 0 on the y-axis up the left, it's percent incidence. So if there were 10 fruit, did we see disease symptoms on how many of those fruit? Okay. Hopefully you can kind of see, I know it's a bit small, but the 4 treatments, again, the 2 varieties and the 2 treatments, the various days that we would take data. And in this case, it's severity. So what percentage of the fruit was diseased? Okay. 0, no spots at all. 100% would be 100% covered. And you see that for the most part, up to that week period, both treatments behave very similarly. As you try to store it longer, I mean, they both start to have problems. Yeah. So the summary for the field, definitely there's a varietal effect, a seasonal effect in either case, with an overall increase in severity. as the season goes on. So you hear how easy it is for these spores to spread once it shows up. I mean, it kind of starts to take over. So really there was no significant difference between the treatments prior to harvest. Okay. For post-harvest, anthracnose was present on all the fruits, regardless of what we were looking at. And I decided to put it in anthracnose because the quotes, that is what we call the Colletotrichum problem, anthracnose. Okay, no significant difference in incidence. So taking the fruit from the field, starting the post-harvest test, you see the percentage of incidence, definitely of the variety effect. And day 7 severity, there's really no difference. So again, shows promise of another option, okay, of this product. But what we really dealt with this last year, we now found these are various symptoms from our post-harvest trials. And we're thinking, well, especially how many Colletotrichum, anthracnose fungi we had collected in about 2021, 2022. Well, we realized we started looking at something else. Not everything was anthracnose, Colletotrichum. We realized we had other genera of fungi. So on that right, the Botrispharia, Neophysococcum, Diaportha, these were other things that we would isolate from some of these symptoms, including some of these Colletotrichum. So it's very important to, like I say, prove, first of all, identify, purify, and prove pathogenicity of what we're looking at. And this is how I'm ending the talk with some of the pictures from our studies. So we did ID. Our lab uses a variety of techniques, including looking at, these fungi under the microscope and using molecular techniques to sequence genes and compare in a database to figure out what species we're actually working with. So we have some names. We then wanted to complete Koch's postulates and went through with both varieties and also different levels of fruit ripeness at color break and mature green. With pathogenicity testing, that's really the process. You start with healthy fruit, a purified fungus, you put them together, you watch and wait, you see what happens, you try to recreate the symptoms we see in the field, then we re-isolate and compare that fungus to what we put on to say, yes, that's what we added, and that actually is the pathogen. Okay, so for the next 4, the bottom left says day 0. This is what it typically looked like at the ripeness level. Okay, then the others are showing the pictures of the different genera back on to try to prove what is actually causing disease and what is not. What's the primary pathogen and potentially what could be these secondary pathogens that I mentioned. Yeah, there might be anthracnose, there's something else. We get it out predominantly. Maybe it's not doing something on its own. Well, this is Raposa. After 7 days, the mature green stage, now what the fruit is, and you can now start to see how these spots are developing, particularly with some of the genera. And we would have the fungal plug and a control plug, okay, so that the process, maybe the small wound isn't doing anything, but it's the fungus added that's actually causing the problem and causing these new symptoms. Okay, so this is the color break stage. Again, what the fruit looked like when we got started and what ends up happening, storing after a week. We kept clearly stumbled upon some new pathogens. This is Manzanillo, day 7. Again, what typical ripeness, day 0, and then after 7 days, what starts to happen with symptom development. Again, very striking, unfortunately. This is the color break stage, and you can see in particular this Neophysococcum parvum and the Botrispharia dotidea definitely have the capacity to be, I mean, they are known, but definitely here for the first time in Hawaii. So for the future plans and kind of wrapping up this grant, it's coming to the end, this overall grant, is to document these new diseases, mango, and see if these essential oils can do anything specifically, kind of those first plate tests and things like that, what it is for these potential new pathogens that might be causing more of a problem, especially post-harvest. Understand better what these essential oils can do, and especially post-harvest. How can we change the system maybe to get better control beyond the week? Definitely share the information as we have it. So with that, I'll say thank you very much. And I'm happy to try to answer any questions now, or there's my email. Definitely drop a note.
Is there a difference in hazards who the guy who's applying the sprays?
No. So that, you mean from the product side?
Yeah.
No, so it's equally as pretty safe and friendly as, the typical grower standard. Again, for organic, the non-organic, it's a whole different ballgame, but again, the label's the law as far as what these products are. But no difference in whatever kind of PPO or application method. Definitely a little pricier. I think that's one of the potential, things to the economic side of it.
Prices between the two are about the same.
Thyme Guard's more expensive. Yep. I'm thinking still it's the application and maybe the length of time from the last treatment to the post-harvest study. We're still more than a month, probably about a month from when the fruit have been last treated to when those post-harvest studies start. So again, immediately the fruit look really nice. It's what happens after that. And if the thought is to try to extend shelf life, I think the application has to be, closer to when harvest occurs.
The both of them, you apply about the same amount once a month?
Yes. Yep.
Sorry, what was the question?
Oh, sorry. Yes, I apologize, people online. Just asking about the overall, PPO requirements, the safety of it's equal in safety as far as Thyme Guard and the copper sulfur, and maybe the cost a little more expensive for Thyme Guard for sure. And I'm sorry, what was your last question?
The amount of spraying once a month?
Yes, sorry. And then the actual number of sprays was once a month throughout fruit development.
How far do you spray until before harvest?
It occurred, so the last spray prior to harvest was almost a month.
Which copper product was it? Pueblo?
No, it's copper. What is
Is it cool side?
it's the number, what copper product is it? No, it's not funny. I'm so sorry. I will definitely get that information to you.
Omni approved?
Yes, it is. Yeah, Omni approved.
Badge, maybe?
It's not funny.
Badges.
Yeah, badges. Yeah, badges.
So the handouts that you guys provided, we have two tables, one in each of the handouts that are this on the side. Are those organic, non-organic?
Yeah.
Yeah, so the question is, there are handouts available. And whether or not, the products are for organic use or not through the handouts.
Yeah,
Yeah.
the tables.
I don't think it specifies organic or not. But we can help you.
What if someone for you, maybe you can help us with that?
It's the copper and the sulfur.
Yeah, beyond copper and sulfur, everything else will be for non-organic use.
Yeah.
I'm just wondering if it was copper hydroxide or copper oxidation or which form of copper is used. It's like three forms of copper and one has.
There's one, it's like, there's a number to it. It's funny. It's more of an ornament, like a copper 29, copper. I'll definitely get you the information. Andrew, are we good on questions online then? Okay, well, thank you, everybody. Appreciate your time. Thank you.
Yeah, it's crazy.
As the activator.
Sorry, it's like.
Oh, what year was that? Is that? Taken. That's a lot.
Was that? Oh, this one, that storm was, that's a Zell. So like 2014? Yeah, that one.
For the most part, you know, the conventional fungicides do a whole lot better.
Yeah, that's what I'm wondering. Well, we'll check with the current. With the current decline, but the other criteria, what you would recommend, because the tree rate, you know, like they don't.
All right. Do you have any more questions? That's it. I think we will call it there. We've got a couple people left in here. So yeah, as Andrew said in the chat, we will send out a link for the recording and post-event evaluation a little bit later today. Thank you guys for joining us and hope you guys had a good time. Learned a couple things. Thanks.